Hello friends. So today we'll be talking about polymerase chain reaction, also called PCR. So the polymerase chain reaction is a molecular technique, or it is an in vitro technique that amplifies specific nucleic acid sequences. Amplify means to make several copies, which could be millions or billions of specific nucleic acid sequences. Let's say, for example, we want to detect the presence of Mycobacterium tuberculosis in the sputum. First of all, the sputum specimen will be obtained, and if this bacterium is present in the sputum, there are specific genomic regions that indicate the presence of this bacterium in the sputum. And these genomic regions have specific nucleic acid sequences. So what PCR does is it amplifies these sequences. But before we go into PCR in details, let us have a basic understanding what the DNA is all about. So the DNA simply means deoxyribonucleic acid. So we need to understand that the DNA is double-stranded. So number one, it is double-stranded. And then the second one is it contains the deoxyribose sugar, okay? The deoxyribose sugar, and then it contains a phosphate group. These two forms the sugar phosphate backbone. So the deoxyribose sugar and the phosphate group bound together forms the sugar phosphate backbone. We have double strands like this. The next one is nitrogenous bases. And the nitrogenous bases includes adenine, guanine. These two are the purines. And then we have the cytosine and thymine. These are the pyrimidines. The adenine pairs with thymine with 2 hydrogen bonds, while the cytosine pairs with the guanine with 3 hydrogen bonds. So these are the hydrogen bonds. So this is the sugar phosphate backbone, and then in between these are the nitrogenous bases. Okay, so if we have adenine, there will be thymine here with 2 hydrogen bonds. If we have cytosine, there will be guanine. So, and then let's say thymine and adenine and thymine, guanine and cytosine. So we can see that both strands are complementary. So we can say this is 5 prime, 3 prime, and this will be 5 prime, 3 prime. That is because both strands are complementary. So if this is 5 prime, this will be 3 prime, and then if this is 3 prime, this will be 5 prime. Now, inside this double helix, okay, are the nitrogenous bases. The next thing that we should talk about are the nucleotides. These are the nucleotides of DNA. So the nucleotide consists of the nitrogenous bases, a phosphate group and sugar molecule bound together. Now, this represents deoxy. Okay, so this small letter D represents deoxy. It shows that it's a deoxyribose sugar. Deoxy adenosine triphosphate. And this is adenine, which represents the nitrogenous base, and then triphosphate. So this is the phosphate group, okay? So adenine bound to a deoxyribose sugar is bound to 3-phosphate group. So this is a nucleotide. So this is a nucleotide for adenine, nucleotide for cytosine, nucleotide for guanine, and then nucleotide for thymidine. So this is deoxy adenosine triphosphate. And then this is deoxycytidine triphosphate. And then deoxy guanosine triphosphate. And then deoxy thymidine triphosphate. So these are the nucleotides of DNA. There are basically two components that are required for PCR. The first is the genetic material. This genetic material is basically obtained from the DNA that has been extracted. I give an example of a Mycobacterium tuberculosis and how its DNA can be extracted and then used for polymerase chain reaction. So let's assume the DNA has been extracted and then we have the genetic material. And then the second thing we need is a master mix. Now, a master mix contains several components, and then this master mix acts as a reagent. So it's just like a reagent. So we can say this genetic material is a sample. Okay? So we can say this genetic material is a sample, and then this is the reagent. Now, the master mix contains some components. So one of those components is Taq DNA polymerase. Now, Taq DNA polymerase is a thermostable enzyme. And what it means is that it has the ability to withstand heat, high temperature. And then it is obtained from a bacterium called Thermus aquaticus. Another component of the master mix is the primer. Now, a primer is a short single-stranded DNA sequences. So we have both the forward and the reverse primers. Now, these primers are designed to be complementary with flanking sequences. So we have something like this. Let's assume this is a DNA strand, and let's assume from this point to this point is where the specific nucleic acid sequence that we need or that is of interest to us are located. So if this is where the specific nucleic acid sequences are located, from this end to this end and from this end to this end are called the flanking sequences. So the forward and the reverse primers binds to the flanking sequences. So let's assume this is the first strand, 5 prime to 3 prime, and then this is the second strand, 3 prime to 5 prime, because they must be complementary with each other. And this is where the specific nucleic acid sequences of interest are located. So from this point to this point and this point to this point are the flanking sequences. So the forward primers will bind there, while the reverse primers will bind there. This strand is called the anti-sense strand, while this strand is called the sense strand. And also, because this is 3 prime, this primer, which is the forward primer, will be 5 prime to 3 prime. And because this is 3 prime here to 5 prime, this reverse primer, this end will be 5 prime to 3 prime, because these primers must be complementary with the sequences here, and these primers must be complementary with the sequences here. The forward primers binds to the flanking sequence, okay, or the flanking sequences of the anti-sense strand, while the reverse primers bind to the flanking sequences of the sense strand. So another component that is required is magnesium chloride. Now, this usually dissociates and to produce magnesium ion, which acts as a cofactor to facilitate the reaction. The next one is the nucleotides. And these nucleotides include the deoxyadenosine triphosphate, the deoxycytosine triphosphate, the deoxyguanosine triphosphate, and the deoxythymidine triphosphate. I gave this during the introductory part of this class. So the nucleotides are also required, where the deoxyribose sugar is bound to the nitrogenous base, which is also bound to 3-phosphate group. So for example, this one is deoxyadenosine triphosphate. So these are also required as components in the master mix. So basically, for polymerase chain reaction to occur, we need the genetic material, which will act as a sample. And then we also require the master mix, which acts as the reagent. But then the master mix contains these components: the Taq DNA polymerase, the primers, which are the forward and the reverse primers, magnesium chloride, and the nucleotides. So there are 3 steps required in PCR. The first one is denaturation. Polymerase chain reaction occurs in circles. Okay? It occurs in circles, and it takes place in an instrument called the thermocircular. Now, what this thermocircular does is, it regulates the temperature of the solution it contains. It raises and decreases the temperature at intervals. So, the genetic material is the sample, and the master mix, which contains all these components, is the reagent. That is to say, an aliquot of the sample will be added into the reagent in a test tube. So let's assume this is the tube. An aliquot of the sample will be added, okay, into the tube, and then a specific volume of the reagent, of the master mix reagent, will also be added to this test tube. And then this tube will now be placed in the thermocycler. So when placed in the thermocycler will begin by raising the temperature of the solution, because the sample and the reagent together is now the solution. So when this solution in the test tube is placed in the thermocycler will now raise the temperature. This denaturation occurs at 95 degrees Celsius. So the thermocycler will raise the temperature to about 95 degrees centigrade or Celsius. And at this temperature, the hydrogen bonds, remember I said that these nitrogenous bases are held together by hydrogen bonds. So what happens is, at this temperature, the hydrogen bonds will be broken. All the hydrogen bonds inside the helix will be broken. And when the hydrogen bonds are broken, the strands will become separated. At 95 degrees Celsius. From this double helix, it will become separated. So this is 5 prime, 3 prime, 3 prime, and 5 prime. Now, after its separation, the thermocycler will also lower the temperature to about 54 degrees Celsius, and that is called annealing. (54 degrees Celsius). Now, when the thermocycler decreases or lowers the temperature to 54 degrees Celsius, that is when the primers will now anneal or bind to their respective strands. And that process is called annealing, because at this temperature, the primers will begin to anneal or bind to their respective strand. So, this, of course, is the forward primer, while this is the reverse primer. So this will be 5 prime, 3 prime, and then this will be 5 prime, 3 prime. And then this will be 5 prime, 3 prime. If this point to this point, this point to this point are where the specific nucleic acid sequences of interest are located, then from this point to this point will be the flanking sequence, and that is where the reverse primer will bind, while the forward primer will bind at this end of this strand. So after this process, the thermocycler will also raise the temperature of the solution to 72 degrees Celsius. Now, at that 72 degrees Celsius, that is where Taq DNA polymerase begins to function, or that is where it is optimal in its activity. So, this process is called extension, also called synthesis. Now, once the thermocycler raises the temperature again to 72 degrees Celsius, the Taq DNA polymerase binds to this end, and also binds to this end. So let's assume this is the specific nucleotide sequence of interest. Okay? What Taq DNA polymerase does is it gets attached here, and then it begins to pick the complementary nucleotide and add. So for this case, since this is thymine, what it will do is it will pick thymine is supposed to bind with what? Adenine. So it is going to pick this. It will now pick this nucleotide and add there. So this will now be what? So let's give a space. Let's say this is 5 prime and 3 prime. Okay, so it will now pick this nucleotide, and then this will be what? Adenine. Then for this adenine too, it will pick this, okay, because it's supposed to bind with thymine. It will pick this, and then this will be what? T. Similar thing, this will be what? G, C, and G. Now, remember that it does not just pick only the nitrogenous bases, it picks the nucleotide. That is to say, it has the sugar molecule attached with the nitrogenous base together with the phosphate group. And I said earlier that this strand is the sugar phosphate backbone. That is to say, because each of these nitrogenous bases has a sugar and phosphate group attached to it, automatically the phosphate backbone will be formed. So automatically, as these nitrogenous bases are adding, the phosphate backbone will be forming. And that is what we call what? Extension. Because this primer has been extended to this point. And that 3' that was located here, as this extension is ongoing, the 3' will also be moving backward until it will get to this point. And this will be what? 3'. Similar thing will occur here. And this will also extend to what? 3'. Now, this is just a circle. This is just one circle of PCR. Now, this circle will continue to even about 30 or 40 circles. In the first circle, one molecule of DNA gave rise to 2. So in the second circle, each of these DNA strands will give rise to what? 2 DNA molecules. Because this one together will unwind, and this one will give rise to 2 DNA molecules. And this together also will give rise to 2 DNA molecules. So we still have what? 2 will give rise to what? 4. So in the first one molecule of DNA will give rise to 2. In the second circle, we have 4. In the third circle, we have what? 8. So basically, we can give it a formula and say 2 raised to power what? N. 2 raised to power n. So the n is the number of circle. So when the number of circle is 1, it will be what? 2 raised to power 1, which is what? 2. When the number of circle is 2, it will be what? 2 raised to power 2, which is what? 4. And when the number of circle is 3, it will be 2 raised to power 3, which is what? 8. And then when the circle is 4, it will be 2 raised to power 4, which is what? 16. So we can say that in polymerase chain reaction, there's an exponential amplification of a specific nucleic acid sequence.